Welcome to the Deep Dive, where we crack open fascinating topics and hand you the most crucial insights. Have you ever paused to truly look at a plant? Perhaps a tiny seedling pushing its way through stubborn soil, or, a fruit ripening on your kitchen counter, changing color and softening seemingly on its own.
Yeah, it's pretty incredible.
It is. How do they know exactly what to do? How do they coordinate all that growth and change?
Well, it's not magic, though sometimes it feels like it. It's actually a hidden language of intricate chemical communication.
Okay.
Today, we're taking a deep dive into the world of plant hormones, these powerful microscopic signals that orchestrate virtually every aspect of a plant's life, from its very first sprout to its final flower.
That's right. And our mission today is to navigate a core chapter on this very topic, distilling the most important knowledge and insights for you. We're basically giving you a shortcut to understanding how plants orchestrate their own lives without getting bogged down in overwhelming jargon.
Exactly. Think of us as your guides, helping you understand the internal factors that regulate all this plant growth and development.
So to really appreciate what these hormones do, maybe we should start by thinking about how plants manage to coordinate everything. Growth, differentiation, development, across all their different parts.
Good starting point. Like animals, plants absolutely need communication between their cells, tissues, and organs. Chemical signals are, well, they're key for that coordination.
So chemical messengers. Now, for many of us, our minds might jump to animal hormones like adrenaline or maybe insulin. Those are typically made in one specific gland. Then they travel through the bloodstream to somewhere else. Is it very different in plants?
There are some really crucial differences, yeah. Plant hormones, or phytohormones as they're called, aren't produced in dedicated glands like in animals.
Oh, okay.
Instead, they can actually be synthesized in multiple locations throughout the plant body. And what's even more fascinating is they can act locally, right where they're made, or they can travel to distant parts. And here's the real kicker. They're active in unbelievably minute quantities, just tiny, tiny amounts.
How tiny are we talking?
Well, imagine a single needle in 20 metric tons of hay.
Wow.
That's roughly the equivalent concentration of a common plant hormone, like indole-3-acetic acid, in a growing pineapple shoot. That's how potent these molecules are.
That's an incredible analogy. It really drives home how a tiny signal can have such a massive impact. Now, the word hormone itself, I think it comes from the Greek hormone, meaning to stimulate, but maybe that's a bit too simple for plants.
It definitely can be, yeah. While many plant hormones do stimulate growth, it's vital to remember they can also inhibit it.
Okay, so not just go, but also stop.
Precisely. And the plant's response isn't just about the hormone's chemical structure. It's about how the target tissue reads its sensitivity. The very same hormone can cause wildly different effects in different tissues or even at different stages of a plant's life. It's context dependent.
So it's less of a simple command and more like a nuanced conversation within the plant. And I think I've heard the term crosstalk used here.
Exactly right. Hormones rarely act totally alone. The huge range of responses we see in plants comes from these complex interactions and crosstalk between various hormones and other signals. Think of it maybe like a whole committee of chemical messengers deciding the plant's next move.
A committee, I like that.
Yeah. And plus, plants can store hormones in inactive forms, often bound to other molecules like sugars, just ready to be released when needed. It's another layer of really subtle control.
So who are these microscopic master communicators then? Traditionally, scientists focused on what they call the classic five, right?
That's right. Auxins, cytokinins, ethylene, abscisic acid, and gibberellins.
Okay.
And more recently, we've formally recognized a sixth major class, the brassinosteroids. These are steroid hormones, and they're essential for normal plant growth.
And there are others too, beyond those main six.
Oh, definitely. There are other important signaling molecules like salicylic acid, which is involved in disease resistance and even heat production in plants like the skunk cabbage,
Right.
and jasmonic acid, which helps plants defend against insects and cope with stress. Plus things like systemin for wound response and even a polypeptide called florigen that triggers flowering. But today, we'll focus mainly on those major six.
Okay, sounds good. Let's start with the very first one ever identified, auxins, often called the growth directors. The story apparently begins way back over 140 years ago with Charles Darwin and his son Francis.
Yeah, 1881. They were doing these really systematic observations of phototropism, how plants bend towards light. They're using young canary grass and oat seedlings.
And what did their experiments actually show?
Well, they noticed something fascinating. If they covered the very tip of the seedling, it's called the coleoptile in grasses with a tiny opaque cap, the seedling stopped bending towards the light.
Okay.
But if they covered the part below the tip or used a transparent cap on the tip, it still bent just fine.
So the signal had to come from the tip itself.
Exactly. Their conclusion was groundbreaking for the time. Some kind of influence was transmitted from that upper part, the tip, down to the lower part, causing the lower part to bend. We now know this bending happens because the cells on the shaded side actually stretch more than the cells on the lit side.
So the Darwins found evidence of this influence, but they couldn't quite pin down what it was.
Right. That took a few more decades. Enter Fritz W. Wendt in 1926. He was the one who successfully isolated this substance from oat seedling tips. He collected it in little agar blocks.
Ah, clever.
Yeah, and he named it Auxin, from the Greek Auxin, which means to increase.
Makes sense. And what is this Auxin, chemically speaking?
The principal natural auxin is indole-3-acetic acid, or IAA. It's actually structurally quite similar to the amino acid tryptophan. And it's mainly produced in actively growing regions. The tips of shoots, the apical meristems, young leaves, root tips, and also developing fruits and seeds. Its role is so fundamental that, interestingly, we haven't found any viable mutants that completely lack auxin. That suggests it's absolutely essential for a plant to even live.
Wow. Essential for life. And you mentioned something earlier. Auxin has a really unique way of moving around the plant.
It really does. Auxin is the only plant hormone known to be transported polarly. That means it flows in a specific unidirectional way.
Unidirectional, how does that work?
So in shoots, it moves downwards from the tip towards the base. That's called basipetal transport. In roots, it's a bit more complex. It moves towards the root tip, acropetal, in the central vascular tissue, and then it gets redirected and moves basipetally away from the tip back towards the base in the outer cell layers, like the epidermis and cortex.
Okay, that's quite specific.
It is, and this isn't just passive diffusion. It's an active, highly regulated process. Auxin moves at speeds of maybe 2 to 20 centimeters per hour, which is much faster than it would just drifting through cells.
So how does the plant manage this precise one-way street for auxin?
It uses specialized protein doors, or pumps, embedded in its cell membranes. There are specific influx carriers that help auxin get into cells, and efflux carriers, like the well-known pin proteins, that control its exit.
Pin protein.
Yeah, pin proteins. And the key thing is that these efflux carriers, these pin proteins, are often concentrated specifically at one end of the cell, so they act like one-way gates, directing the flow.
Like bouncers at a club door, only letting people out one way.
Exactly. And what's really cool is the plant can dynamically move these pin proteins around in the cell membrane. They cycle in and out. This allows the plant to actually change the direction of auxin transport in response to signals like light or gravity.
That is sophisticated. So auxin really is like a director organizing traffic flow. What are some of its key roles in guiding how a plant develops?
Well, one absolutely crucial role is in differentiating vascular tissue, essentially designing the plant's internal plumbing system, the xylem and phloem.
How does it do that?
Gradients of auxin concentration influence how these water and nutrient-conducting cells develop. For instance, in a young leaf, auxin flowing from the tip induces the formation of the main central vein. Then, as the leaf expands, the auxin flow shifts towards the margins, inducing the network of lateral veins.
That's amazing. It's like drawing the lines on a map.
It is. And if a stem gets wounded and the vascular bundles are cut, applying auxin above the wound can actually induce new vascular tissue to differentiate and bridge the gap, reconnecting those vital pathways. It's also important for connecting the vascular traces from leaves into the main stem bundles.
That makes me think of a common gardening practice when you pinch off the top bud of a plant to make it bushier. Is auxin involved there?
That is a perfect classic example of apical dominance. The auxin that's produced in the growing apical bud, the very top tip, flows downwards. And this stream of auxin strongly inhibits the growth of the lateral buds, the ones sitting in the leaf axils along the stem.
So it tells the side shoots, not your turn yet.
Pretty much. So when you pinch off that shoot tip, you remove the primary source of that inhibitory auxin. The auxin flow diminishes, and that releases the lateral buds from inhibition. They're then free to grow out, making the plant bushier.
And you can even prove that.
Yeah, you can actually apply an Auxin paste, like lanolin paste containing IAA, to the cut surface where the tip was, and it will mimic the effect of the intact bud, keeping those lateral buds suppressed. It's quite neat. And interestingly, there's another hormone, a more recently discovered one called strigolactone, that seems to work together with auxin to fine-tune this whole process of apical dominance.
Okay. What about roots? Does auxin help with root growth too? I mean, people use rooting hormone when they take plant cuttings, right?
Exactly. That rooting hormone you buy is typically a synthetic auxin. Auxin is a key signal for forming lateral roots, the ones that branch off the main root. And it's absolutely crucial commercially for initiating adventitious roots. Those are roots that form from non-root tissues, like a stem cutting.
So it helps start roots.
Yes, but it's a bit of a delicate balance. While auxin promotes the initiation of new roots, very high concentrations can actually inhibit the elongation of already established roots. So context and concentration are everything.
Right, the dose makes the poison, or the promoter.
Precisely. Auxin also plays a really significant role in fruit development. It's often produced by the developing seeds inside a fruit, and this auxin promotes the growth of the surrounding fruit tissue.
Ah, so the seeds tell the fruit to grow.
In many cases, yes. And you can even trick the plant. Treating unpollinated flowers with auxin can sometimes lead to parthenocarpic fruits, basically, seedless fruits. Think of some varieties of tomatoes, cucumbers, or eggplants. There's a classic experiment with strawberries, described in the book. If you remove all the tiny seeds, the little Auxins, from the surface of a developing strawberry, the fleshy part, the receptacle, stops growing.
Okay.
But if you then apply auxin to the surface, growth resumes. It shows the seeds were the source of the auxin needed for the fruit to develop.
That's a clear demonstration. Now, how can something that promotes growth also be a weed killer? That seems contradictory.
It does seem odd at first glance, but synthetic auxins, like the well-known 2,4-D, are widely used as herbicides, particularly effective against broadleaf weeds, leaving grasses relatively unharmed.
Why does that work?
Unlike the plant's natural auxin, IAA, which gets broken down fairly quickly, these synthetic versions are much more resistant to degradation by the plant's enzymes. So when they're applied, they build up to artificially high, frankly lethal, levels inside the weed.
An overdose.
Exactly. This massive overdose completely disrupts the plant's normal hormonal balance and growth processes, essentially causing it to grow itself to death, while the more resistant crops, like corn or wheat, are less affected.
Okay, that makes more sense. A powerful tool, but also needs careful use. All right, let's move on. Next up, let's talk about cytokinins, often called the cell division promoters. The discovery story here sounds fascinating, involving coconut milk.
It's a fantastic story, really highlights scientific curiosity. Back in the 1940s, a researcher named Johannes van Overbeek found that coconut milk, the liquid endosperm inside a coconut, contained some unidentified but very potent growth factor that could really spur the growth of plant embryos in culture.
Just from coconut milk?
Yeah.
Yeah.
This finding spurred a lot of research. Skoog and his colleagues at the University of Wisconsin were working with tobacco stem tissue in culture. They found that the tissue would enlarge, but it wouldn't really divide and proliferate unless they added something else besides auxin.
They needed another ingredient.
Exactly. Eventually, Carlos O. Miller, working with Skoog, isolated a highly active compound from autoclaved herring sperm DNA, of all places.
Herring sperm DNA. Okay, that's unexpected.
Right. They called this compound kinetin. It wasn't naturally occurring in plants in that form, but it strongly promoted cell division. Later, Miller and others isolated the first naturally occurring, highly active cytokinin from immature maize kernels, or corn. They named it zeatin.
Zeatin from zea maize, the corn plant. Makes sense.
Precisely. And because these compounds were so effective at promoting cytokinesis, the actual process of cell division, they collectively named this class of hormones cytokinins.
So cytokinins are key for cell division. Where do you find them in the plant, and what else do they do?
Chemically, they are derivatives of adenine, one of the DNA bases. They're abundant in actively dividing tissues. Think root tips, shoot tips, young leaves, developing seeds and fruits. They are often synthesized in the root tips and then transported upwards to the shoots through the xylem, the water-conducting tissue. Though locally synthesized cytokinin is also really important, for instance, in helping buds break dormancy.
And they interact with auxin, right?
Very much so. Their ratio with auxin is critical, especially in laboratory settings like tissue culture. If you take a lump of undifferentiated plant cells, called a callus, and you give it a high auxin to cytokinin ratio, it tends to form roots.
Right.
But if you give it a high cytokinin to auxin ratio, it tends to form shoots or buds. If the concentrations are roughly equal, it often just keeps growing as an undifferentiated callus. Figure 2710 in the book shows this really clearly with tobacco callus.
So you can basically dictate development by tweaking the hormone balance.
In many cases, yes, it's a powerful tool. And in the intact plant, cytokins generally promote the growth of lateral buds, effectively acting as antagonists to Auxin's apical dominance effect we discussed earlier. They also seem to negatively regulate lateral root formation, kind of doing the opposite of Auxin in that context.
And they have another interesting role related to aging.
Yes, delaying senescence. Cytokinins can postpone the aging and yellowing of leaves, which happens when chlorophyll breaks down. Florists actually use synthetic cytokinins sometimes to help keep cut flowers and foliage looking fresh for longer.
Well, that makes sense.
There's a neat experiment described where if you dab a spot on a detached cocklebur leaf with kinetin, that spot stays green while the rest of the leaf yellows. Even more, radioactively labeled amino acids applied elsewhere on the leaf will migrate towards that cytokinin-treated green spot, suggesting the cytokinin helps maintain protein synthesis and nutrient import.
Fascinating. Okay, moving on to our next player, ethylene. This one's a bit different, isn't it? It's a gas.
It is. Ethylene is a simple hydrocarbon, C2H4, and it's a gas at normal temperatures. And yes, it has a bit of an infamous history. Its effects were noticed long before it was identified.
How so?
Back in the 1800s, in German cities using coal gas for street lighting, people noticed that shade trees growing near leaky gas mains were defoliating prematurely, dropping their leaves.
Leaky gas pipes were hurting the trees.
Exactly. It wasn't until 1901 that the Russian scientist Dmitry Neljabov identified ethylene, a component of that coal gas, as the culprit. He showed that even incredibly low concentrations, like 0.06 parts per million, could cause weird growth effects in dark-grown pea seedlings. They'd get short and fat and grow sideways.
Wow. So where does the plant make this gas itself?
Plants synthesize ethylene from the amino acid methionine. There's a well-defined pathway involving intermediates called SAM and ACC. Figure 2711 outlines this. And crucially, ethylene production often ramps up significantly in response to various stresses like drought, flooding, injury, and also in response to high auxin concentrations and during processes like fruit ripening and senescence.
It's like a stress signal, but also involved in normal development.
Very much so. It has some really remarkable and diverse effects. One classic example is the triple response that Neljabov first observed in those dark-grown pea seedlings if you expose them to ethylene.
The short, fat, sideways growth.
Exactly. As shown in figure 27 to 12, ethylene causes three things. Decreased longitudinal growth, they stay short, increased radial expansion of the stem, they get thicker, and horizontal growth, they grow sideways instead of straight up.
Why would a plant do that?
Well, think about a seedling trying to push up through soil that might have obstacles, like a rock or compacted layer. Growing short, thick, and sideways could help it navigate around that obstacle to reach the surface and light. It's an adaptive response.
Ah, that makes perfect sense. Clever adaptation.
Ethylene is also crucial for survival in some flooded conditions. In semi-aquatic plants like deep-water rice, rise in floodwaters trigger ethylene production, which then causes rapid stem elongation, allowing the plant to keep its leaves above the water surface.
Amazing.
And even in non-aquatic plants, ethylene can help submerged tissues survive by promoting the formation of air spaces, allowing oxygen to diffuse down from the parts still above water.
But the big one people probably know ethylene for is fruit ripening, right?
Absolutely. This is where ethylene really shines in the popular imagination. It plays a crucial role in the ripening of what we call climacteric fruits. These are fruits like apples, bananas, avocados, tomatoes. Figure 2713 shows the spike in ethylene and respiration during ripening in a tomato.
Climacteric. What does that mean?
It means these fruits show a dramatic increase in respiration rate called the climacteric rise, coinciding with ripening. And this whole process is triggered by ethylene. Ethylene sets off a cascade of changes. Chlorophyll breaks down, so fruits change color. Enzymes digest pectin in the cell walls, making the fruit softer. And complex carbohydrates and acids are converted into simple sugars, making them sweeter.
All the things that make fruit appealing to eat.
Exactly, which, from the plant's perspective, makes them more attractive to animals who will eat them and disperse the seeds. And ethylene production itself is autocatalytic in these fruits, meaning a little ethylene triggers the fruit to make even more ethylene, leading to that rapid ripening phase.
That's why one bad apple spoils the bunch. Or one ripe banana ripens the rest.
Precisely. The ethylene gas released by one ripening fruit signals to its neighbors to start ripening too. And this is used commercially all the time. Fruits like bananas or tomatoes are often ticked green and hard for shipping, and then exposed to ethylene gas at the destination to ripen them uniformly before they hit the supermarket shelves.
Very practical. Does ethylene do anything else?
Yes, it also promotes abscission, the shedding of leaves, flowers, and fruits. It triggers the formation of an abscission layer at the base of the petiole or peduncle, where enzymes dissolve the cell walls, allowing the organ to detach cleanly.
So it helps plants drop old leaves or ripe fruit.
Right. And this is also used commercially. Ethylene-releasing compounds are sometimes sprayed on crops like cherries or blueberries to loosen the fruits, making mechanical harvesting easier. It can also be used for thinning fruits on trees like prunes or peaches, ensuring the remaining fruits grow larger.
And one more role mentioned is sex expression.
Yeah, this is pretty neat. In plants like cucumbers and squash the cucurbits, ethylene plays a major role in determining whether a flower develops as male or female. Higher levels of ethylene tend to promote femaleness, often by inducing programmed cell death of the male parts, stamens, in flowers that start out as potentially bisexual.
Wow, hormones controlling flower sex. Okay, let's shift gears again. Abscisic acid, or ABA, often called the dormancy inducer and water saver. The name sounds like it should be involved in abscission, but you said ethylene does that.
Right, there's a bit of historical confusion there. In the late 1940s, Paul Waring found a growth inhibitor accumulating in dormant buds, which he called dormin. Then, in the 1960s, Frederick Adicott's group found a substance in cotton leaves and fruits that accelerated shedding, or abscission, and they called it abscission.
Two different discoveries.
Initially thought so, but then they discovered that dormin and abscissin were chemically identical, so the name abscisic acid, or ABA, stuck. However, further research showed that ABA's direct role in causing abscission is actually pretty minor in most cases. Ethylene is really the primary driver there. So the name ABA is a bit of a misnomer in that respect.
Okay, so despite the name, it's not the main abscission hormone. What are its critical functions then?
ABA is hugely important, primarily for inducing and maintaining seed dormancy and for mediating the plant's response to water stress.
Let's start with dormancy. Why is that important?
Well, you don't want seeds germinating at the wrong time, right? Like still attached to the parent plant or just before winter hits. ABA levels typically increase during early seed development. This ABA stimulates the production of storage proteins for the embryo, but crucially, it also prevents premature germination, keeping the seed dormant until conditions are favorable, maybe after a period of cold or when there's enough water.
So it acts like a wait signal for the seed?
Exactly. And declining ABA levels, or decreased sensitivity to ABA, are often linked to the breaking of dormancy and the start of germination. There's a striking example in the book May's Mutants called viviparous mutants. Vivipary means germinating while still attached to the parent. These mutants either can't synthesize ABA properly or they're less sensitive to it, so their kernels sprout right there on the cob. Figure 2715 shows this.
Wow, that really shows ABA's role in keeping seeds dormant. And you said it's a water saver, too. How does it help with drought?
This is another major function. ABA acts as a crucial root-to-shoot signal when the plant experiences water stress. When the soil starts to dry out, the roots sense this and begin to synthesize more ABA.
Okay.
This ABA is then released into the xylem, the water transport stream, and moves rapidly up to the leaves.
And what does it do in the leaves?
In the leaves, ABA acts on the guard cells that surround the stomata, those tiny pores on the leaf surface that regulate gas exchange, but also water loss through transpiration. ABA causes these stomata to close.
Ah, plugging the leaks.
Precisely. By closing the stomata, ABA significantly reduces water loss from the leaves, helping the plant conserve precious water during drought conditions. You can really see the importance of this if you look at certain wilty mutants plants that have a genetic defect preventing them from making ABA. Their stomata tend to stay open even when water is scarce, so they wilt very easily and often can only survive in very high humidity environments.
So ABA is critical for drought survival. Okay, next on our list. Gibberellins, or GAs, the height enhancers and germination triggers. This discovery has another interesting origin story, right? Something about foolish seedling disease.
That's right. This story takes us to Japan in 1926. A plant pathologist named E. Kurosawa was studying a disease affecting rice seedlings. These infected seedlings would grow excessively tall and spindly, become weak, and often fall over before they could produce grain. The farmers called it bakane, or foolish seedling disease.
Foolish because they grew too tall too fast.
Exactly. Kurosawa discovered that the cause wasn't the plant itself, but rather a substance produced by a fungus, Gibberella fujikuroi, that was infecting the rice. He found he could apply sterile filtrates from this fungus to healthy rice seedlings and induce the same symptoms.
So the fungus was making a growth promoter.
Yes. It wasn't until 1934 that two other Japanese scientists, Yabuta and Sumiki, finally isolated and crystallized this substance and named it gibberellin after the fungus. Quite a surprising origin for what turned out to be a major class of natural plant hormones.
Absolutely. So what exactly are these gibberellins and what do they do in plants?
Chemically, they're part of the terpenoid pathway, like ABA. Over 136 different naturally occurring gibberellins, designated GA1, GA2, and so on, have been identified from plants, fungi, and bacteria, though only a few seem to be biologically active as hormones in plants themselves. GA3, also known as gibberellic acid, is one of the most studied and commercially available.
And where are they found?
They're synthesized in various parts of the plant, but the highest concentrations are typically found in young tissues of the shoot, and especially in developing seeds.
Okay, and their effects, height enhancement.
Yes, that's one of their most dramatic effects. Gibberellins strongly stimulate stem and leaf elongation. They do this by promoting both cell division and cell elongation, or stretching. The effect is most striking in certain dwarf mutants. If you take a genetic dwarf plant that's short specifically because it lacks gibberellins or can't respond to them, and you treat it with GA,
It grows tall.
it often grows to a normal, tall stature. Figure 2717 shows this beautifully with dwarf pea seedlings treated with GA. This understanding of gibberellins and dwarfism was actually crucial for the Green Revolution in agriculture.
How so?
Well, traditional tall varieties of cereals like wheat and rice often lodged, meaning they fell over in wind or rain, especially when heavily fertilized. Breeders selected for semi-dwarf varieties, often carrying mutations affecting GA synthesis or response. These shorter, sturdier plants could handle more fertilizer and put more energy into producing grain instead of straw, leading to huge yield increases.
That's a massive impact. What about germination?
Yes, gibberellins are also key players in breaking seed dormancy and promoting germination, often acting counter to ABA. In some species, applying GA can substitute for environmental cues like cold stratification or light exposure that are normally required to trigger germination. It essentially signals the embryo to start growing.
And there's a specific example in barley, related to beer.
Ah, yes. The barley example is classic. It's fundamental to the malting process used in brewing. When a barley grain starts to germinate, the embryo synthesizes and releases gibberellins. These GAs diffuse to a specialized layer of cells surrounding the starchy endosperm called the aileron layer. You can see this in figure 2718. The GAs then stimulate the aileron cells to synthesize and secrete various hydrolytic enzymes, most famously iamylase.
And iamylase does what?
It breaks down the stored starch in the endosperm into simple sugars. Other enzymes break down proteins. These sugars and amino acids then provide the food the growing embryo needs to develop, shown in figure 2719. It's a beautiful system where the hormone triggers the mobilization of stored food reserves. And interestingly, ABA has the opposite effect. It inhibits the GA-induced synthesis of iamylase.
A nice example of hormonal antagonism right there. Do GAs do anything else?
Yes, a few other notable things. In some plants that normally grow as a compact rosette of leaves near the ground, like cabbage or spinach, gibberellins can induce bolting, that's the rapid elongation of the flower stalk, and subsequent flowering, often substituting for a required cold treatment or long day photo period. Figure 2720 shows this.
So they can trigger flowering in some cases.
In certain types of plants, yes. And like Auxin, gibberellins can also induce the development of parthenocarpic or seedless fruits in some species. Commercially, they are hugely important in viticulture, especially for growing table grapes like Thompson's seedless. Applying GAs makes the individual grapes larger and also elongates the stems within the cluster, creating looser bunches that are less susceptible to fungal diseases. Figure 2721 shows the dramatic difference.
Very useful commercially. Okay, that brings us to the sixth major class. Brassinosteroids. These are the newest additions to the main group.
Relatively speaking, yes. Although growth-promoting substances from pollen were reported back in the 1970s by J.W. Mitchell and colleagues, they called them brassins because they first found them in pollen from rape plants, Quercus canapus. It took until 1979 to isolate and identify the most active compound, brassinoide.
Brassinoide. And what kind of hormone is it?
It's a polyhydroxylated steroid hormone, chemically distinct from the others we've discussed, but like GAs and ABA, it's derived via the terpenoid pathway. Since then, about 70 related compounds, collectively called brassinosteroids, have been found in virtually every part of the plant. Figure 2722 shows the structures. Unlike some other hormones that travel long distances, endogenous brassinosteroids seem to primarily act locally near where they're synthesized.
And are they really essential?
Absolutely essential for normal plant growth and development. This was clearly shown by studying dwarf mutants of the model plant Arabidopsis that were defective in either synthesizing brassinosteroids or responding to them. These mutants are severely stunted, with smaller leaves containing fewer and smaller cells.
So without them, plants are tiny.
Exactly. And conversely, if you genetically engineer plants to overexpress genes involved in brassinosteroid synthesis, they often grow larger than normal.
What specific roles do they play?
They influence a really wide array of processes: cell division, cell elongation in both roots and stems, vascular differentiation, branching, lateral root development, seed germination, leaf senescence. Even responses to light and resistance to various stresses. They seem to be involved in almost everything.
A jack-of-all-trades hormone.
You could sort of say that. One particularly well-studied role is in the differentiation of tracheary elements, the water-conducting cells, xylem. There's a neat experimental system using isolated mesophyll cells from zinnia leaves, figure 2723. These cells can be induced in culture to de-differentiate and then redifferentiate into functional tracheary elements. It turns out brassinosteroids are required for the final stages of this process, including the proper formation of the secondary cell wall and the programmed cell death that hollows out the cell.
So they're crucial for making the plant's pipes work properly.
In essence, yes. They ensure those final maturation steps happen correctly.
Okay, we've met the major players and seen their diverse effects directing growth, dividing cells, ripening fruit, managing stress. But how do these tiny molecules actually do all this? How do they create such big changes at the cellular level?
That's really getting to the heart of it, the molecular language. Fundamentally, hormones act as chemical messengers between cells, coordinating things like when and where cells divide, and when and how much they expand or change shape. They exert their influence primarily by affecting gene expression.
Gene expression, switching genes on and off.
Exactly. Remember, virtually every cell in a plant contains the same complete set of genetic information that's the principle of totipotency. But obviously, not all genes are active in all cells all the time. Hormones are key signals that tell specific genes within a cell's nucleus when to switch on or switch off.
How do they flip those switches?
They typically interact with or trigger pathways that lead to the activation or repression of regulatory transcription factors. These are proteins that bind to specific sequences on the DNA near a gene, controlling whether that gene gets transcribed into RNA and ultimately translated into a protein.
So the hormone signal gets translated into a change in protein production.
Precisely. We saw that with the barley aileron cells. Gibberellin activates the transcription factor that switches on the allelase gene, while abscisic acid somehow represses it. Modern techniques like microarrays show that applying a single hormone can alter the expression levels of literally hundreds, sometimes thousands, of genes within hours. Table 27-2 in the book gives some examples of genes regulated by different hormones.
Wow, massive reprogramming. But plant growth isn't just about making new proteins, it's also about cells physically getting bigger, right? It's cell expansion.
Absolutely. Cell expansion is fundamental, especially in plants where cells are confined by rigid walls. How much a plant cell expands depends on two main things: the internal hydrostatic pressure, called turgor pressure, pushing outwards against the cell wall, and how much the cell wall itself can stretch irreversibly, its extensibility. Think of it like inflating a balloon inside a cardboard box. The pressure is the air, the box is the wall.
Okay, turgor and wall stretchiness. Figure 2724 illustrates this, I think.
Right. Hormones primarily influence growth by altering the cell wall's extensibility. Generally speaking, auxin and gibberellins tend to increase wall extensibility, loosening the wall and allowing the cell to expand under turgor pressure.
Making the box slightly stretchier.
Exactly. Conversely, hormones like abscisic acid and ethylene often decrease wall extensibility, making the wall more rigid and thus inhibiting growth. There is a classic idea called the acid growth hypothesis, suggesting auxin works partly by activating proton pumps that acidify the cell wall space, which in turn activates enzymes that loosen the wall structure.
Okay, so they control how much a cell expands. But what about the direction? How does a cell grow long and thin versus round and fat?
That's determined by the architecture of the cell wall itself, specifically the orientation of the cellulose microfibrils within it. These are like strong reinforcing rods. If these microfibrils are deposited randomly, the cell tends to expand equally in all directions, becoming spherical.
Okay.
But if the microfibrils are laid down primarily in parallel hoops, oriented transversely around the cell, kind of like the hoops around a barrel, then the cell can stretch much more easily longitudinally, parallel to its main axis, than radially. It gets longer, not wider. Think of stretching a coiled spring. It gets longer easily, but it's hard to make the coil itself fatter.
Right. The hoops resist sideways expansion.
Precisely. And guess what influences the orientation of these newly deposited cellulose microfibrils? Well, indirectly. The orientation of the microfibrils is guided by microtubules, which are protein filaments lying just inside the plasma membrane. Figure 2725 shows this relationship. And hormones can influence the arrangement of these guiding microtubules. For example, gibberellins often promote a transverse arrangement of microtubules, leading to transverse cellulose deposition and thus longitudinal cell elongation, making stems grow tall.
Okay.
Ethylene, on the other hand, as we saw in the triple response, can cause microtubules to reorient longitudinally, leading to more random or longitudinal cellulose deposition, which allows for radial expansion, making stems shorter and thicker.
So hormones control the cell's internal scaffolding, which dictates the wall's architecture, which determines the direction of growth. That's intricate.
It really is. A whole cascade of control.
So we have the hormone arriving at a target cell. How does the cell actually perceive that signal and initiate these changes in gene expression or cell wall properties? It sounds like a relay race inside the cell.
That's a great analogy. It involves signal transduction pathways. The hormone, the initial signal, first binds to a specific receptor protein. This binding event triggers a chain reaction, a series of biochemical events inside the cell that ultimately leads to the final response. Much of what we know about these pathways comes from studying mutants, particularly in the model plant Arabidopsis, that are insensitive to a particular hormone or show a response even without the hormone.
Can you give an example? How does ethylene signaling work?
Sure. The ethylene pathway is quite well understood, partly because it's a bit unusual. Ethylene receptors, like one called ETR1, are actually located on the membrane of the endoplasmic reticulum inside the cell. Now, here's the twist. In the absence of ethylene, these receptors actively stimulate a negative regulator protein called CTR1. CTR1 then keeps the downstream pathway switched off.
So the receptor normally puts the brakes on.
Exactly. But when ethylene gas binds to the receptor, it inactivates the receptor. This stops the receptor from stimulating CTR1, so CTR1 becomes inactive. This releases the brakes on the next protein in the chain, EIN2. Activated EIN2 then somehow transmits the signal to the nucleus.
Okay.
In the nucleus, the signal leads to the activation of transcription factors like EIN3 and ERF1, which then switch on the specific genes responsible for the ethylene response, like the triple response genes. Figure 2727 diagrams this cascade. It's a bit counterintuitive, with the signal working by inhibiting an inhibitor.
Release of inhibition. Okay. What about other hormones?
The pathways for auxin and gibberellins share some similarities. They both involve receptors located inside the nucleus. When the hormone binds, it essentially causes the degradation of specific repressor proteins that were previously sitting on DNA and blocking gene expression. Removing the repressor allows the genes to be switched on.
So again, removing a brake.
Exactly. Though the precise mechanism differs slightly, auxin acts like a molecular glue, helping its receptor bind to the repressor to target it for destruction. Gibberellin binding causes a shape change in its receptor, which then allows it to interact with the repressor.
And cytokinins and brassinosteroids.
Cytokinin signaling is more like some animal hormone pathways. The receptors found in Arabidopsis are transmembrane protein kinases located at the cell surface. When cytokinin binds, the receptor adds phosphate groups to itself, autophosphorylation. This phosphate group is then passed down a chain of other proteins, like a bucket brigade, eventually reaching and activating a transcription factor in the nucleus.
A phosphorylation cascade.
Precisely. Brassinosteroid signaling also involves a receptor at the cell surface called BRI1. Figure 2728 shows this. It works together with a co-receptor called BAK1. Hormone binding brings them together and activates BRI1's kinase activity, again initiating a phosphorylation cascade inside the cell that ultimately alters gene expression.
So lots of different strategies, but the end result is often changing which genes are active. You mentioned amplifying the signal, too.
Yes, signal amplification is crucial. A single hormone molecule binding to a receptor needs to trigger a large enough response inside the cell. This often involves second messengers.
Second messengers.
These are small molecules or ions inside the cell whose concentration changes rapidly and transiently in response to the initial hormone signal, the first messenger. A classic example is calcium ions, Se2 plus serlone. Hormone binding might trigger channels to open, causing a temporary spike in the cytoplasmic calcium concentration.
Okay.
These calcium ions can then bind to and activate various downstream proteins, like protein kinases. Since one kinase can phosphorylate many target proteins, this significantly amplifies the original signal.
Turning a whisper into a shout.
A good way to put it. And sometimes the hormone response isn't about changing gene expression, but a more direct physiological effect. The ABA control of stomata is a perfect example.
Right, you mentioned that earlier. How does that work at the molecular level? Figure 2729 helps here.
It does. When ABA arrives at a guard cell, it binds to receptors. This binding triggers the opening of calcium channels in the cell membrane and also possibly internal membranes, causing that influx of Cy2+ we just talked about. Calcium is a second messenger here.
Okay, calcium floods in.
This rise in cytoplasmic How2+ then triggers other channels to open, specifically channels that let negatively charged ions like chloride and malate flow out of the guard cell. This outflow of negative charge changes the electrical potential across the membrane, which in turn causes potassium channels to open, letting positively charged potassium ions flow out as well.
So the cell loses a lot of dissolved stuff, solutes.
Exactly. The loss of all these solutes, calcium, chloride, malate, potassium, makes the water potential inside the guard cell less negative, or higher. Water then flows out of the guard cell by osmosis following the solutes. This loss of water reduces the cell's turgor pressure.
It goes limp.
Essentially, yes. The guard cells lose their rigidity, and the stomatal pore between them closes up. When the ABA signal disappears, the ions are actively pumped back into the guard cells, water follows, turgor increases, and the pore reopens. It's a direct, rapid physiological response, mediated by ion channel activity, all triggered by the hormone.
That's a beautifully orchestrated cellular dance, all to save water. What an incredible journey into the intricate lives of plants.
It really is, isn't it? We've seen how these 6 major groups of plant hormones, auxins, cytokinins, ethylene, abscisic acid, gibberellins, and brassinosteroids, along with other signaling molecules, act as the microscopic conductors of plant life.
Yeah, conductors is a good word.
They orchestrate just about everything, from a seed's very first sprout to a fruit's final sweet ripeness. And they don't work in isolation. They're constantly interacting in complex combinations, responding dynamically to signals from within the plant and from the surrounding environment.
So next time you see a plant, maybe a towering tree or just a small houseplant on your windowsill, it's worth considering that silent, incredibly complex chemical conversation happening constantly within its cells.
Absolutely. It's a conversation that allows that plant to adapt, to grow, to respond, and ultimately to thrive in its world, shaping its very existence from moment to moment.
It really makes you wonder, what else might we learn by listening more closely to this intricate biological language?
Yeah.
What other secrets are plants holding?
A truly thought-provoking question to end on, don't you think? Food for thought.
Absolutely. Thank you so much for joining us on this deep dive into the secret world of plant hormones. We hope this exploration has given you a newfound appreciation for the hidden wonders happening inside every plant around you. Until next time, keep exploring.